Please go ahead, take the floor.
All right, thanks everyone for coming to our webinar on normative modeling and single subject neuroimaging analysis. I guess that a lot of you perhaps heard a lot about normative modeling at the OHBM Global in Glasgow just a couple months ago. So today we are very glad and very pleased to have 3 lined up international and Aussie-based speakers. They're all ECRs. They did wonderful work, so they are going to share their wonderful work and hands-on tips on how to do normative modeling and single subject neuroimaging analysis. So I hope that after our webinar, a lot of you will develop some new interest in normative modeling or know better how to do normative modeling yourself. So I hand over to Johanna to introduce our first speaker.
Yeah, hello everyone from me as well. I'm very pleased to introduce our first speaker, Sage Rutherford. Sage is a PhD candidate at the Donders Institute in Nijmegen, the Netherlands, working with Dr. André Marquand on machine learning applications in psychiatry. Prior to her PhD, she studied biophysics and computer science, worked as a research assistant collecting infant functional MRI in Detroit, Michigan, and as a data scientist in the Department of Psychiatry at the University of Michigan. Outside of science, she spends time with her dog, Charlie Mop, and doing fun sports like hula hooping, yoga, and stand-up paddleboarding. We're very much looking forward to your talk, Sage.
Hi, it's nice to be joining you from the Netherlands. Let me just share my screen. Okay, are we seeing not presenter mode? Okay, so yeah, today my talk is titled Evidence for Embracing Normative Modeling. And I'm going to present the first chapter of my PhD work and a few new results at the end. So I would like to begin by acknowledging that all of this work is really done on behalf of the lab that I'm a part of. Normative modeling has been around for several years. Andre's lab's really done a lot of work. There's a lot of great papers published before I joined the lab, and I kind of came into the lab 2 years ago and really took on the project of making the toolkit for normative modeling, building better documentation, writing different tutorials on it, and making it a little bit more accessible. So I just want to acknowledge that this is, everything I'm presenting is very much a group effort. So just to introduce myself a little bit, my research interests are really kind of centered between machine learning on one hand. My background is very technical, but all of my research experience has been working with clinicians. So I really try to think about balancing these two domains. So one of my advisors, Andre, really is an expert in machine learning, and another advisor, Eric, is a psychiatrist who spends most of his time actually in the clinic with patients. So then there's me in the middle, and I see myself kind of bridging this gap and thinking about machine learning for healthcare, and most specifically in psychiatry. So just want to highlight the 2 kind of papers that I'll be presenting today. They've both been published now. I call them together practical considerations for normative modeling. On one hand, we have a paper published in eLife, and this is what I consider a data set paper where I've pre-trained a lot of normative models and share them and share code for using those pre-trained normative models. And the second is just recently came out in Nature Protocols and is a tutorial paper where we really walk you through start to finish, how to prepare your data for normative modeling, how to think about the different choices when setting up a normative model, and how to run it. So a lot of what I'm presenting today are figures and work from these papers, but obviously I only have 20 minutes, so check them out for more details. Okay, so to zoom out a bit, I just want to talk about why we might need normative modeling. So often a method that we think about is comparing a group of healthy controls and a group of clinical subjects. Maybe this is people with major depressive disorder, but we're really relying on this clinical label. And we compare the 2 groups and look at the mean. And this often results in kind of no group differences. And this is because we're only looking at mean effects, which requires this consistent spatial signature within each group, and then that spatial signature to differ between groups. And when we look at the underlying data, we see that this isn't really the case, and across groups and within group, there's a lot of variance that we're not really properly capturing and modeling, and it's represented, a lot of patients, there's not this consistent spatial signature, so we're not finding these group effects. So realizing this has kind of led us to, okay, we need to look at things other than just the mean and group differences, and that led me to realizing that normative modeling was a good approach for doing that. So like I said, normative modeling has been around for a long time outside of even just neuroimaging. It's most widely used maybe in pediatrics when you look at, say, a child is 2 years old and it's a girl and she's this height or weight, and you can then look at where she is compared to the population and see if she's above or below and if there needs to be some intervention. And this panel is just showing kind of what I mentioned on the last slide, the assumptions of group difference testing, where you have a group of controls, a group of clinical, and it assumes that there's some clear separation between the groups, which in reality, there's a lot of overlap between groups, and we're not able to capture and separate that with our group difference testing, but normative modeling is really designed to do that. So in normative modeling, we have a set of clinical variables such as age, sex. We usually try to represent MRI scanner site because we're often working with multi-site data, sometimes some kind of set of data quality metric. And then we're going to build a type of linear regression. In this work, I'm using a Bayesian linear regression, but there's many different algorithms that you can use, and we'll predict some brain variable. So far, I've been really focusing on cortical thickness because it's just very widely available. But there's, you can use function or other metrics as well. So just to really quickly cover some of the steps involved in normative modeling, we first have to think about our data selection. And this is things like what type of imaging modality you want to use. What is the image resolution? Do you want to model at the ROI level, at the vertex or voxel level? What type of covariates do you want to include as predictors? Typically, we just use things like age and sex because that's what's widely available across sites. But there's also starting to be some interesting work using things like cognition or psychopathology. And you're not really limited into what your covariates can be. It's whatever fits your research question. You next need to think about, and this is something that really needs to be carefully decided upon, is your inclusion criteria for the normative sample. So this is the sample that you'll be measuring deviations against. And so you have to, really carefully think if this sample matches, the sample you're trying to represent. Because if they're very demographically different, then your test sample, you're going to detect a lot of deviations, but it might not be for the reason that you think you. So the next, you have to prepare your data. We have a nice Python toolkit, the PCN toolkit, which stands for Predictive Clinical Neuroscience, name of Andre's lab. You have to do a little bit of pre-processing, a little bit of quality checking, train and test splitting, and setting up some Python environments. But like I said, we have tutorials that really walk you through each of these steps if you're not familiar with Python or the toolbox. Next, you have to think about what type of algorithm you want to use to run the normative model. A lot of earlier work focused on Gaussian process regression. In this work, I've used Bayesian linear regression, but in the paper, we walk you through some of the other choices. And the last step of normative modeling is evaluation and interpretation. And when you run a normative model, you depending on the input data resolution, whether it's ROI or voxel, you have kind of high resolution data. And then what is output from a normative model is also that same resolution. So for every model, you have a deviation score, and you have to then think about how to summarize that. So that's what happens in this step. And it's also, you have to think about how to evaluate, see if the model is fitting well. So And in this work, I also, along with the papers, created a website on Read the Docs and some nice Python notebooks that can be launched directly in your browser using Google Colab, which is a free service that is really nice for kind of sharing scripts and things. So yeah, there's just a lot of resources, and these are all the tutorial links as well. Okay, so to the big data, like kind of normative model paper I've shared, we started with all of these large data sets. There's such a plethora now of data that's been shared. Our training sample was around 60,000 subjects. And with this in mind, we wanted to be able to train a normative model on this sample so we could capture population variance in cortical thickness data. But we then wanted to be able to transfer this and show that we could transfer this to smaller sites that were collected maybe in a clinical setting. And yeah, our goal of this initial work was really to focus on this transfer. So yeah. A hurdle to some of the widespread application of normative modeling is kind of that there haven't been these well-defined reference models. And there's been a lot of work on it, but people haven't really been sharing these pre-trained normative models. So that was another goal of this work. And we thought that these models had a few requirements. They should accurately model population variance across large samples. They should be derived from widely accessible measures. Provide the ability to be updated as additional data come online, be supported by easy-to-use software tools, and they should quantify brain development and aging at a high spatial resolution so that the patterns can be used to stratify cohorts and predict clinical outcomes. There has been a little bit of other normative modeling work, like sharing pre-trained models, but they've really focused on more global representation, such as total gray matter volume. But if you're interested in a psychiatric application, you kind of need a little bit more spatial precision than just total gray matter volume. You might be more interested in a specific region, for instance. So in the eLife paper, we had a couple different test sets to show kind of really how well these models were performing under a lot of different restraints. Our initial sample was about 58,000 subjects, ages 2 to 100 from 82 different sites. And we split that into training and test sets, so almost 30,000 subjects across each train and test. We then had another testing set, which is called MQC, or manual quality checking. When you start working with very large data sets, it becomes hard. You have to rely on automated quality checking metrics, which is great, but there's kind of always this thought in the back of your mind, what if some of our data is bad? So in a smaller subset, but still quite large, of 24,000 subjects, all of these subjects had been visually inspected for quality, their free surfer data and their underlying T1 data. Our next test set was a clinical test set. This was transdiagnostic, included schizophrenia, depression, social anxiety disorder, autism, and ADHD, almost 2,000 subjects. And we had a transfer set, which was never included in any of the training or initial testing. And it was showing that we could really transfer our model to new sites that were not included in the testing. And that was almost 600 subjects. So this is just a distribution of all the different sites and ages across all of the testing and sets I showed you in the last slide. So showing that we kind of have a nice spread of the different ages. This is initial results of that model in the healthy controls test set. We're explaining quite a bit of variance across the cortex and subcortex, up to almost 60% variance. And around the circle here, we're just seeing some different trajectories. Okay, so quickly go through this. These are just some of the metrics we use to evaluate normative models. Explained variance is kind of really easy to interpret. The skew and kurtosis show the Gaussianity of the deviation scores, so we would expect this to follow a normal distribution. And across all different test sets, the main takeaway from this is that our evaluation looks fairly similar. So I was especially excited to compare the full test set to the manually quality checked test set, and really excited to see the results holding that they were performing very similarly. So we wanted to further evaluate the models and show across sites. So in this slide, I was collapsing across all of the sites. We wanted to see, do these results hold up across all of the different sites, across all of the different test sets. And I'm just showing you the explained variance here. And the takeaway is kind of that the distribution across all of the sites looks pretty similar. And we're, it's not like there's a few sites maybe that, maybe we're performing a little bit worse at, but relatively consistent. And that's just showing the age distribution again across the sites. So it's not, our performance of the model isn't really correlated to the age distribution of the site. So that says even if you have, kids at one site or very old at one site or people in their 40s at one site, our model isn't dependent on that. So that was nice to see as well. Okay. Next, I want to show just a couple slides of some work I'm really excited about. All of the work I just showed was sort of how to do a normative model, how to evaluate it, but then we're still at this point where, okay, I've fit a normative model, what do I do now? And there's a lot of things that you can do with the outputs or the deviation scores from normative modeling. So I've tested a couple different benchmarking sets. One is where I do a classification of healthy controls for schizophrenia. That's what I'm showing on this slide. And I compare using the deviation scores versus just using the raw cortical thickness data as inputs to this logistic regression model. And what I found were that using the just the cortical thickness, the raw data that hadn't been input into the normative model, our classification rate was essentially chance, 50%. But if I use the deviation scores from the normative model, our accuracy goes up to 0.72, which is still maybe low, but there's a really significant increase. And this was using a sample of roughly 260 healthy controls and 260 schizophrenia patients. So this was really exciting to see. It shows that normative modeling is sort of revealing some of the important clinical variation that might have been, without the normative model, maybe the site or age or sex variation was getting in the way of detecting this. We also, beyond just classification, looked at predicting general cognitive ability in the Human Connectome project. And we used, again, deviation scores in one predictive model and just the raw cortical thickness data in the other predictive model. And we tried several different algorithms for setting up the prediction of the cognition. And we see across using all of the deviation scores compared to the cortical thickness, our accuracy, which is established by the mean squared error, is always lower for using the deviation scores compared to using the cortical thickness, with the exception, I think, of the ridge regression. But for most of the other algorithms, again, using deviation scores leads to a better prediction of cognition. So that was also very exciting for me to see. And the last test we did was just going back to the original kind of case that I said we shouldn't be doing, but if we just do kind of our classical group difference testing, but the input to that is the deviation scores from normative modeling or the raw data, what we find is on the top, I'm showing the effects of the deviation scores comparing healthy controls versus schizophrenia. And on the bottom, I'm showing just the raw cortical thickness data. And the yellow areas are non-significant. We don't find any group differences. And the dark blue areas are FDR corrected p-value, so significant group differences. And the takeaway is really that when you use deviation scores in this analysis, there's a lot more significant results that are revealed. And again, this is kind of follows the same story as the classification, using the normative modeling scores, kind of reveals the important clinical variation that we care about that's maybe hidden if we don't properly model it. So these results are coming out in a paper soon. I know this was really quick overview, but hopefully I gave you enough information to be excited about it. And I would like to end again by just acknowledging the wonderful lab that I'm a part of, and also my dog, Charlie Mop, because we could all use more dogs and sunglasses in our life. And with that, I'm happy to answer some questions.
Thank you, Sage. That was wonderful. Really interesting. Let me see. We have a first question from Andrew Zaleski. He says, an AUC of 0.5 is surprising for the case of raw phenotypes in schizophrenia classification. Was age and sex regressed out before features were used in the classification experiment? Previous work predicting schizophrenia diagnosis using cortical thickness have achieved AUCs well above 0.5.
Yeah, that's a great question. In the plot that I showed, it was not regressed out. I used age, sex, and site as an additional predictor, but I am, that analysis is a little bit of a work in progress, and I'm currently looking at, if I refit that model, residualizing for age, sex, and site instead of just having them as predictors. But I was also surprised by it.
Great. There is another question from Sina. He says, amazing body of work on normative modeling. You mentioned that there are various models, Gaussian process regression, Bayesian regression, GAM-LSS, to perform normative modeling. I wonder, what is your take on the benefits and pitfalls of different approaches? Why would a model be selected over another?
Yeah, that's a great question that comes up a lot, and I dive a lot into detail in the Nature Protocol paper, but Gaussian process was what was used initially when we were training these models on smaller sample size, think like less than 100 subjects. And it was really effective, but it does not scale that well when you start training models of thousands of subjects. So that's when we started using some of the Bayesian regression models. So I chose Bayesian linear regression in this. It was fitting well, and it runs very quickly. The hierarchical Bayesian regression model is a really great choice. It allows you to more flexibly, fit the models. You can, with the Bayesian linear regression, site is a fixed effect, and you can just fit additional parameters in the HBR, but the BLR and HBR are pretty similar. The BLR is just faster, and in the case of cortical thickness, the evaluations were really good. The explained variance and all the other metrics were fitting really well, so I just chose to stick with the BLR. But if the models were not fitting very well, the HBR allows you to just tune a lot of additional hyperparameters. So if you need to change the shape of the model or anything, the HBR just has a lot more for you to tune than the BLR, but it's a little bit slower. So that was really my main choice.
Great. Thank you. I think in the interest of time, we might actually move on. Thank you so much, Sage. That was wonderful.
Thank you for the opportunity. It was really nice to be here and to talk with all of you guys.
And thank you for getting up so early. Bye.
Thanks, Sage, and thanks, Johanna. So I'm going to introduce our second speaker, which is from, she's Ashley Siegel. So Ashley is currently completing her PhD in psychiatry and neuroscience from Monash University under the supervision of Professor Alex Bonado, Dr. Kevin Aquino, I hope I pronounced correctly, and Dr. Lyndon Parks. So her research focuses on understanding the neuroheterogeneity of mental illness across various brain scales. I'm very looking forward to your talk today. Ashley, if you're ready, can you share the screen?
Great, thanks very much for the introduction. Great, I'm assuming you can see my slides without the presenter view.
Yeah, correct. Looks perfect.
So thanks very much for the organising committee for the opportunity. It's quite a privilege to be able to present this work that forms part of my PhD, hoping that I can inspire at least one person in the audience of why normative modelling is cool. But before I dig in, I would also like to acknowledge the people of the Kulin Nations on the land in which we meet today. I pay respect to the elders past and present and extend my respect to Aboriginal and Torres Strait Islander people joining us today in this webinar. I note that sovereignty was never ceded. It always was and always will be Aboriginal land. All right, so to get things started, hopefully what I will present today will be complementary to some of the stuff that Sage shared with us, and hopefully not too much overlap, but still inspiring all the same. So as Sage nicely documented, that there's been thousands of neuroimaging studies that have documented diverse structural changes associated with psychiatric disorders. Despite this, neurobiological mechanisms remain poorly understood and clinically useful biomarkers are lacking. One reason for this is what Sage alluded to, is this standard case control design, which focuses on a comparison between group mean values and ignores the considerable heterogeneity observed in patients. While this perhaps was a reasonable assumption in the early stages, there are many possible scenarios of why this isn't the case, with heterogeneity widely reported in patients. For me, this is why normative modelling offers a really exciting opportunity to really dig in and understand these individual differences associated with psychiatric disorders. Like Sage beautifully explained, it is like the growth chart that you've seen in your doctor's office. However, instead of your height, you would look at the grey matter volume, where you get these normative estimates of grey matter volume as a function of age, and then you can make predictions about individuals, with the idea that patients might deviate from this normative range, but the way in which they deviate would vary. So here it would indicate either significant increases or decreases in grey matter volume. This work has been applied across a whole range of different psychiatric disorders to date, with the predominant finding is that patients typically have more deviations, as you would predict, compared to healthy controls. However, they're highly heterogeneous in their anatomical presentation. So the specific location of these deviations varies considerably, such that no more than 20% of patients have shown a deviation in the same anatomical lesion. When you consider that majority of studies to date have based their understanding of psychiatric neurobiology on this case control design, in contrast to normative modelling literature that has found that the group mean is not representative of an individual, I think this really offers quite an exciting opportunity for us in the field at the moment to dig in and really ask these, to use these new frameworks to ask really interesting questions. One question that I'm particularly interested in is that despite this biological heterogeneity often reported by normative modelling, patients with a given condition still show a level of phenotypic similarity that is sufficient to warrant a common diagnosis, raising an important question. If patients with the same diagnosis show minimal overlap in their individual patterns of brain deviations, what explains this similarity in clinical presentation? So if we think back to Sage's presentation, we can consider these two individuals as patients with a diagnosis of schizophrenia. They both have deviations from this normative range, as demonstrated here in orange. However, the location is different between the two individuals. We can consider the brain as a connected network and that those pathological processes can spread to affect distributed interconnected systems, meaning that it is possible for the deviations in diverse regions to impact the function of common connected areas, such as those that are ballooning out to you here in yellow. This principle has been demonstrated through lesion network marking studies, which has shown that patients with the same neurological syndrome, such as hallucinations, often show very little overlap in the anatomical location of their lesions, but the lesion sites are often functionally coupled to common upstream or downstream sites. For us, this approach has not yet been applied in psychiatry because traditionally individuals with a psychiatric disorder don't exhibit overt lesions. However, they do present with deviations as demonstrated in the normative modelling literature. So we sought to bring these two techniques together to gray matter volume variations associated with psychiatric disorders and the functional circuits within which they're embedded in the following 6 different psychiatric disorders. Importantly, this work would not have been at all possible without open source collab and our dear collaborators. It's a multi-site study consisting of more than 25 different scan sites. For the purpose of this talk, I'll only talk about the negative deviations or significant reductions in grey matter volume, but in the preprint that I'll share with you later, we also report the positive deviations or increases in grey matter volume. So if we just focus on this regional level for a moment, and specifically within the normative modelling framework. As Sage has nicely introduced the concept, hopefully I can be quite brief in the way that I work through it, but feel free to ask questions if I skim over anything. So we first acquired T1 scans from around 2,000 individuals, and we used voxel-based morphometry to obtain grey matter volume estimates. We then parcellated the data using the Tian subcortical atlas and the Shaper 1000 atlas. From here, we then split the data into 3 groups. The first is our training group. This is used to estimate the normative model as a function of age, sex, and site. For the purpose of our work, we use hierarchical Bayesian regression because of the multi-site nature of our data. So the control group consisted of only healthy controls, and then we made predictions about an independent clinical group, about our independent clinical group, and then we also made predictions about an independent healthy control group. The reason of why we included this independent healthy control group will hopefully become clear as I work through the methods further down. Once you obtain, from here, you can then obtain a deviation Z-score map, which tells us for any given brain region how much an individual deviates from this normative range of functioning, as indicated here. Where a negative deviation or a reduction in grey matter volume is signaled in red, where a positive deviation or an increase in grey matter volume is signaled here in blue. From here, what's typically done in the normative modelling literature is a threshold is applied to identify extreme deviation maps or regions that are associated or perhaps atrophied or associated with a given disorder or an individual. For the purpose of our work, we applied a Z-score of less than -2.6. Using these extreme deviation maps, you can then ask the question of what's the consistency within a given group. So is the pattern of deviations that are present for a particular clinical group homogeneous, or are they quite different? So we did this both for our healthy control group and for each of our patient groups, with a darker color here indicating greater consistency in the overlap of extreme deviations. This is what's typically reported in the normative modelling literature to date, but we wanted to go one step further and ask, well, what is the difference in overlap between these 2 groups, between each clinical group and the healthy control group? Do patients with a disorder show significantly greater overlap relative to controls? To do this, we pulled out this difference in % overlap for each clinical group and the healthy control group. And then we used standard permutation testing where we randomly assigned group labels to form our null distribution. We then compared this null distribution with what we observed in the empirical data to identify those regions that show significantly greater overlap in patients. What we found across each of the 6 psychiatric disorders that we studied was that very few regions showed significantly greater overlap in patients relative to controls. This is signaled by the dark pink or red here after correcting for multiple comparisons. Of these significant regions, the maximum overlap for any given group did not exceed 6%, suggesting that no more than 6% of patients showed overlap in any given brain region. This provides support for the early work of normative modelling and our hypothesis that, at least at a regional level, psychiatric disorders are associated with widespread heterogeneity. So building on this work, we then wanted to look at the circuits within which these regional deviations lie, with the hypothesis that although patients with a common diagnosis might be highly heterogeneous in their regional presentation, perhaps they lie within common circuits. To understand this, we first looked at the extreme deviation maps that we again obtained from the normative model for both for each case group and our control group. Then for each individual deviation within for a particular individual, we pulled out the seed-based functional connectivity map using a normative connectome from the human connectome resting state connectivity dataset. We did this for every single deviation for a particular patient or healthy control. And then we obtained the union map, which tells us all the regions that are functionally connected to the deviations associated with that individual. This can be obtained for all the cases and all the controls. We can then follow a similar path as what we did before to look at the percentage overlap between these two groups. However, we were again not interested in just the percent overlap, but whether there was significantly greater overlap in regions functionally connected to the heterogeneous brain regions associated with the clinical group compared to the controls. We did this using two nulls, the first being a group-based null, and the second was a spatially constrained null. I'll walk through the importance of each and hopefully give you a bit of insight of why both of those were critical to evaluate what's really going on. So the first was the standard group-based null, which is almost exactly the same as what we did in the first level of the regional analysis, where we had the percent overlap maps for both our case and control. And then we randomly shuffled, we used standard permutation testing where we shuffled the group labels to identify those regions that show significantly greater overlap in patients. What we found was that all disorders showed some evidence of greater circuit level overlap, with areas of the frontal, parietal, and insular cortex being implicated across most conditions, with cross-disorder difference being more about degree rather than kind. This is demonstrated if you look at the top two rows of ADHD and autism, for example, with very few regions implicated compared to disorders like depression and schizophrenia, where almost the entire brain is implicated. Further, across each disorder, the spatial overlap at the circuit level ranged between 30 and 55% for a given brain region. This compares to just 5% of what we observed at the regional level. However, one challenge with this null-based approach is that, as you would predict, and as what I've indicated throughout a couple of times within this talk, is that individuals in the clinical groups typically have more deviations, as you would expect, than the control group. So what we wanted to ask is, what about this deviation burden? Are the results that we're seeing at this group level approach simply driven by a deviation burden as opposed to something specific to a given disorder? This is because individuals with disorders have more regions implicated in their functional connectivity union map, since the map will be constructed from a larger number of seed-based functional connectivity analysis, therefore increasing the chance of observing overlap across individuals. On one hand, this higher circuit level overlap has real phenotypic consequences, since a higher deviation burden is an expected characteristic of psychiatric disorders, and they're likely to have an impact on behavior. However, on the other hand, it is important to know whether the overlap is simply driven by these group differences in deviation burden, or whether it reflects some preferential targeting of a circuit within a given disorder. To disentangle what this means, we therefore evaluated the statistical significance of the circuit level overlap using a second spatially constrained permutation test. This involved evaluating group differences with respect to a null distribution that preserves the number of deviation-related FC maps contributed by each individual, thus matching the differences in the total deviation burden. What this looks like is that if we go back to our early step of normative modeling, where we obtained these Z-score deviation maps for both our case and control group, we then use spin tests to preserve the spatial order correlation, but to shift the location of where the deviations lie. We then can apply the same threshold as we did earlier on to identify surrogate extreme deviation maps. Importantly, the number of deviations are exactly the same within the null distribution as what is in the empirical data. However, the locations have been randomly shifted throughout the brain. From here, we can follow the same analysis as what I outlined before to find these union functional connectivity maps, to then look at the difference in percent overlap between the two groups within the null distribution. This can be carried out 10,000 times to form our null distribution to compare to what we observed in the data. This analysis therefore tests whether the observed group differences in the circuit level overlap are greater than expected for the functional connectivity maps generated with the same number of randomly chosen seeds. Using this null, the prefrontal regions were identified as showing greater overlap compared to controls in all clinical groups. However, only one region in depression in the prefrontal cortex and 2 regions in bipolar survived multiple comparison corrections. Both these two, the prefrontal cortex has been strongly implicated in both depression and bipolar and has been used as a target site to treat depressive symptoms. So our body, the work that we've done really supports this existing body of literature that there's something important to the prefrontal cortex, at least in the expression of depressive symptoms. More generally though, this result suggests that many of the case control differences that we did observe at this circuit level are associated with total deviation burden, as opposed to some preferential accumulation of deviations in a particular circuit for a given disorder. To an extent, this challenges the existing body of literature that suggests that dysfunction in a specific circuit is associated with a specific disease or disorder. Instead, we suggest that a disorder is associated with complex changes that affect diverse neural systems, often transdiagnostically. So to wrap up, at a regional level, using normative modelling, we observed this extreme heterogeneity of grey matter volume deviations and that it's a general characteristic of mental illness. At a circuit level, we found that these deviations were often coupled to common functional circuits and networks, with the prefrontal and parietal circuits across disorders may be a marker of transdiagnostic distress. More broadly, our findings underscored the need to consider the network in the context of disease-related psychopathology and indicate that the clinical expression of disease alone is not driven alone by primary pathology. More broadly, our findings really show some cool work that you can do with normative modeling generally and utilizing multiple tools with open data. Finally, I'd like to end by acknowledging the wonderful people and data that made this work possible and invite you to ask any questions.
Thank you so much, Ashley. This is amazing and very thorough investigation. I'm so enjoyed. I think we have some questions, but Marcus, would you like to speak up or would you like to type? But I don't know how to make you speak. Okay, we have here. In a way, your findings suggest that there are many possible ways for patients to be different. Do you agree? In that case, do you think your findings, I don't know, so this question is still not completed yet, but like, let's ask the, maybe answer the first one. Do you agree that many possible ways for patients to be different?
Absolutely, I agree.
Yeah, okay. Do you think your findings may reflect different causes, bio, psychosocial, family history, or other factors that might relate to definition of how to be defined like patients abnormal?
Yeah, absolutely. And I think that's one of the really exciting areas of this research. Obviously, it's relatively new, this idea of using normative modeling to study individual differences. And it's really been quite broad brush of looking at the link between how you model deviations in relation to age. I think a lot of value will come from looking at a symptom-based approach or relating this to clinical data more broadly. But I think it particularly is a challenge if you're using open data, because it's quite hard to harmonize data and have clinical data that's available across multiple sites. That's common.
Thank you, Ashley. Any other people have questions? Okay. If you have any question, please just post in the Q&A. And in the meantime, actually, I have a quick question just while waiting. So I saw your work, instead of looking at global measures, like global brain measures, you look at regional heterogeneity. I say that the brain atlas you use has quite high resolution relatively. So would you think the regional measure would be potentially more noisy and less reliable when we build the normative models in comparison to those global measures? And how would you address that? I mean, what's your thoughts on that? In terms of why you choose this particular resolution of this brain atlas?
Great question. I think so. We tried to find a balance between too coarse and too fine, if that makes sense. So instead of taking the whole brain or voxel level, we tried to find somewhere in the middle. But I think if you look at different levels of analysis, you're likely to see less. So if you look at more fine-grained, I think you're likely to see less overlap, where if you look at more coarse-grained, you're more likely to observe greater overlap using normative models. I don't think there's a perfect answer for which level you should use. I think probably the best answer is to apply analysis like this across multiple scales and multiple parcellations and find that see if it reproduces, and if it doesn't, why might that be?
Yes, true. Yeah, so it's a quite challenging area, I know. You have so many options for brain analysis, and we need to keep going doing the research. And I have comments from Michael. So Michael Breakspear, so great talk, Ashley. Can you please post a link to the preprint? Post in chat, or somebody already have that. So please just copy that and paste in the chat. Anyone have any final question? We have one final chance before we move on to our third speaker. If no, Johanna? Okay, great. So your turn, Johanna. Let's introduce our third speaker. Thanks again, Ashley. It's a wonderful talk.
Thank you.
Yeah, thank you, Ashley. So I have the pleasure to introduce our third speaker, Dr. Varda Sayeda. She's the lead of preclinical neuroimaging at the Melbourne Neuropsychiatry Centre, where she contributes to a multidisciplinary team investigating healthy and perturbed mouse brain development from adolescence to early adulthood, with a focus on mouse models of schizophrenia. Additionally, Dr. Sayeda actively collaborates with other researchers at MNC and beyond to facilitate clinical integration of advanced research in neuropsychiatric disorders, such as autism, schizophrenia, and schizotypy disorders in children. She has a background in electrical and biomedical engineering and provides expert contributions in methods development for advanced multivariate image processing and inference. Very warm welcome to Varda, and the stage is yours.
Thank you, Johanna. So I will now share my screen. Okay. Hi. Can you see the presentation in the proper view?
Yes.
Yeah. Perfect. All right. So today I'm going to talk about affinity scores, which is an individual-centric fingerprinting framework for neuropsychiatric disorders. So Ashley and Sage have really set up the stage for me in terms of, what are the considerations from a clinician point of view for individuals with neuropsychiatric disorders. And some of the clinical priorities that lie are not to identify the key symptoms and psychosocial factors at an individual level to facilitate accurate diagnosis, as well as we need to identify the comorbidities that might be existing so that we can have an accurate prediction of illness prognosis at an individual level on the spot in clinics. So all of this is really challenging in neuropsychiatric disorders because, as we know, that there is overlap between symptoms of many disorders, and then there is also a whole lot of variability and heterogeneity within the spectrum of symptoms of specific disorders. So to address these challenges, we propose the use of affinity scores, which is designed to characterize individual patients and to aid in timely diagnosis of psychiatric disorders. So the affinity scores are a little bit deviation from the normative modeling that we have been hearing about today, but they can actually work really well and in complementary way to the information that we get from normative model. And I will go into more details about how that can be achieved. So again, this is a very individual-centric approach. So we want to break away from the conventional population-centric or mean-centric case control design type studies. And using the affinity scores, we can address within order heterogeneity, as well as if there are overlap across disorders at an individual level, and also if an individual is an outlier. So we are actually able to integrate that information into their affinity score and bring that to the attention of the clinician. So it's a complementary way of measuring deviation without having to rely on a reference population. So now to take a bit of a look under the hood, like how exactly are we achieving this? So the affinity scores can actually be calculated both at a univariate and multivariate level. And the underlying premise is quite simple. So because we know that there is so much overlap across distributions of different groups, across any biological measure, such as brain volume or regional brain volumes. So the idea is that why don't we only then look at the neighborhood of an individual? So only look at the information which is potentially most relevant to an individual's profile. So the way we do this is that the first step is to standardize all the measures. So everything is converted into standard deviation units using z-scores, for instance. And then we take each individual one by one, for instance, the blue individual here that we see, and then define a neighborhood around it using a fixed hop size. Now, what is important here is that this hop size in the affinity scoring framework is automatically selected based on the effect sizes which are present in the distributions across various groups. So in this pictorial representation here, for instance, we say that maybe there are 3 representative groups within the data set that we have. So once we have defined a neighborhood around a participant, then we actually look at the group composition. So how many individuals with a particular group are then actually falling into the neighborhood of this participant, and then use that information to calculate the affinity score. So therefore, if an individual is an outlier or an atypical individual, so this could be because of the inherent property of the disorder or the prevalence, or it could be because of the lack of data. So if there is insufficient data, all of that could be implicitly taken into account. And again, we define the neighborhood using the fixed hop size that was determined on the distribution characteristics across the groups, and identify the group level composition just in the neighborhood there. So using this for each variable, so we start by variable by variable, and then we calculate a multi-dimensional affinity score where the dimension of the score will be determined by the number of the representative groups which are present in the data. And then we determine the proportion of neighbors from each group. And this score is then also adjusted by the group level sample sizes, as well as the probability of finding a particular neighborhood. So this way, once the affinity scores have been identified, and we have identified the closest participants, which could be coming from different groups in the population, we can actually calculate a number of further multivariate metrics to help us then performing analysis that can be determined based on the question that we are asking. So I cannot go into detail of all the multivariate metrics that we are calculating, and there is a very detailed description of these in the paper. However, I can give an example. So for instance, we can calculate the common neighborhood-based affinity metric, which basically tells us that in how many number of variables there are a pair of participants which are sharing a common neighborhood. So for instance, in the data set, if there are 2 individuals which are very close to each other, then we would expect that they would be sharing these neighborhoods across multiple variables. And this way we can actually quantify how much overlap there is. So in a way, we can visualize it as finding a partial neighbor, depending on how many variables they are sharing a neighborhood in. So we implemented this framework, the affinity scoring framework, on the Australian Schizophrenia Research Biobank data, just as a proof of concept to see whether just using this localized information across each participant, are we able to determine their individual level fingerprint that can then be informative in a clinical setting. So here is an overview of the data. So in this data set, we have individuals with chronic schizophrenia, as well as treatment-resistant schizophrenia, where treatment resistance is defined as if the individuals are being treated by clozapine. And then we also have data from healthy controls. So because there is no restrictions as such as to the number of variables that can be included in the affinity scoring framework, because we are calculating the scores in each variable separately at first, we did include a range of variables covering the cognitive domains, the clinically relevant information, as well as the brain structure measures such as regional volumes. So here is a sample of affinity profile, how it can look like, for instance, if we are looking at the structure variables. And here we are seeing the signature coming from a healthy participant, a schizophrenia participant, and a TRS participant. So I will unpack the information that we are seeing here a little. So the colors are represented. So the blue colors are representing control, the red are schizophrenia, and the purple is TRS. So now, for instance, if we look at the signature of the healthy participant, we can see that there are some regions which are red. So what it means is that although this is a healthy participant, within that particular brain region, it had more neighbors from schizophrenia group than compared to healthy controls or TRS groups. So similarly, there are some regions which are blue, meaning that this particular individual shared the greatest number of neighbors coming from healthy group, and so on and so forth. So now all this is well, but we actually then wanted to have a mechanism of testing then the statistical significance of having this distribution of neighborhood based on group membership. So the way then we do that could actually be using non-parametric testing, such as permutation testing, to find out what is the probability of having this particular group composition in the neighborhood of a particular individual. And also we can do bootstrapping, for instance, to determine confidence intervals across the affinity scores. So here at the graphs at the bottom, the circular graph at the bottoms are again just showing just a subset of subcortical regions as a proof of concept. So the shade of the color then basically is the log p value. So the darker the color means that the less probability of finding this particular composition of group membership in the neighborhood of this particular individual. And then the confidence intervals are given as the bars. So this way, just at a glance, a clinician is actually then able to determine, okay, what is the overlap of this individual with other groups, representative groups in the population and in each variable separately, and whether it has any statistically relevant information in this neighborhood composition or not. So similarly, these are some profiles from cognitive variables that were available in the ASRB data set. And again, we can see, so the length of the bars are telling us about the strength of the affinity, for instance, and we can see, for instance, in this schizophrenia, patient with schizophrenia, like in their LNS and attention tasks, for instance, they actually had very healthy, so more healthy membership in the neighborhood of this variable compared to others. But then on some of the variables, such as the variables which are measuring their IQ and stuff, we see that they are actually more likely to have TRS or schizophrenia-like signature and so on. So for each variable level, we're actually then able to determine this, composition of degree of overlap with other groups. So similarly, there's some some evidence or fingerprint from clinical data that was present in the ASRB. Now, based on this proof of concept, so I just wanted to emphasize that these affinity scores can actually be used in a number of ways. And the two examples that I wanted to touch on today are the first concept is identification or diagnostic verification, and the other one is classification. So within the diagnostic verification, we actually allow the participant to be included as part of their own neighborhood, so they can actually vote towards themselves. Whereas in the classification framework, the participants are excluded from their own neighborhood, and the neighbors are actually then determining the label for that particular individual. So first, I will give an example of how the affinity scores can actually be used to, determine classification or a clustering-like approach, where we can then aim to identify, for instance, if there's a subgroup of data which are exhibiting a particular phenotype or disorder using the affinity scores. So what we are seeing here is basically what we did for the ASRB data for this classification framework was to relabel the TRS individuals as chronic schizophrenia. So we had the grouping of either healthy controls or schizophrenia, which combined both the chronic and the treatment-resistant schizophrenia into a single diagnostic group. Then we calculated the affinity scores and implemented an affinity score-based classifier to then assess the classification separability of individuals with TRS. And here are the results. So after the classification was done, we relabeled the individuals with TRS to their original label to see how their scores actually performed. So on the graph here on the left, we can see that the individuals with TRS actually showed low affinity to both the healthy controls and individuals with schizophrenia, as well as the graph on the right is again showing that the TRS individuals were not showing affinity again to either schizophrenia or healthy controls based on the affinity scores. So this is as a proof of concept that we can actually use these scores as features then to identify various subpopulations within the data. So another kind of, this is an ongoing work, and I just wanted to demonstrate how the affinity scores can actually be integrated into a clinical decision support system to help the clinicians on the spot about, making inferences about the diagnosis of individuals. So this work was implemented on the data from 85 children from the OS's inpatient ward, and I am collaborating with Dr. Michelle Adams from Monash University on this data. So she's been wonderful and has really input to how we can make this stuff more clinically relevant. So these children were coming from 3 groups. So sorry, I should have written it here, but you can see in the graph. So they are coming from ADHD, ASD, autism, and anxiety disorder. So these are the 3 representative groups which we have in the data. And all the patients are aged less than 12. And we wanted to kind of see whether using affinity scoring system, we are actually able to achieve diagnostic verification for these kids or not. And so far, we have assessed the data from 50 kids clinically. So Michelle actually looked at their clinical files to see whether affinity scores are actually helpful in, picking up the nuances of the diagnosis for these children. So out of those 50 kids, 43 actually retained their original diagnosis. And here is an example fingerprint, for instance. And I must mention here that this fingerprint was actually calculated using the raw clinical data. So no pre-processing was done on it to identify just the neighborhoods and see how the neighborhood composition looked like at the level of each variable. So what we are seeing here on the graph are the affinity scores, as well as in the brackets, we can actually then see the actual test score for this particular individual on the autistic screening questionnaire, for instance, as well as the size of the neighborhood that was defined across it, and then how many individuals were actually present there. So this way, we are actually able to then drill down to an individual level signature to see how it works. So out of the 7 children that did not achieve diagnostic verification using the affinity scores, I would just like to share the case study of one of those kids. And so just a context that this was a child that had an initial diagnosis of anxiety and a subsequent autism diagnosis. And the problem was that the clinician, the treating physician thought that perhaps this child also has ADHD, which is the main underlying cause for the functional distress that this child is experiencing. However, the ADHD was not diagnosed at this stage. So we use the clinical data from this individual child, and what came out was that this child actually showed highest affinity to the data of other children from ADHD, followed by anxiety and autism. So this is where I guess it becomes a bit apparent of the nuanced nature of how affinity scores can actually be helpful in the clinical settings, that it did actually still retain the diagnosis for anxiety and autism that this child previously had. However, it gave like a intuitive and quantitative ranking of what could be causing the primary distress in this child. And so subsequent to that, actually not subsequent, in parallel, Michelle had already delivered a clinical reassessment for this child, and the gold standard test for ADHD actually validated this additional diagnosis, and then our treatment plan was subsequently designed accordingly. So with this, I would just like to conclude my talk and also acknowledge the contribution of various researchers that has gone into making this work possible.
Thank you, Warla. That was beautiful, super interesting. I would encourage people to write their questions in the chat. We might have time for 1 or 2 quick questions, although we're kind of a bit tense with time. There is a question from Sambras. I have if a specific measure, a cognition variable, for instance, from healthy control is showing high affinity, strong neighborhood with schizophrenia patients, does that mean that this measure can identify preclinical healthy individuals, or does it mean that the cognition measure is not reliable to differentiate between healthy controls and schizophrenia groups?
Yes, excellent question. So it can actually mean both things, right? So this is where I thought that it is really important to then have this statistical significance. So for instance, if we are seeing that, such a composition where a healthy control is actually showing more affinity to schizophrenia patients, maybe that particular composition of the neighborhood is not statistically significant. So this is where permutation testing can actually be used then to have this indication of how important or how unique, I guess, this for this particular individual, this neighborhood composition is. If we, for instance, find out that, yes, it is actually really stable and very less likelihood of finding this particular composition for this particular individual, then that could be interpreted as a preclinical marker of some schizophrenia-like association in this healthy individual. So this is where affinity score can actually really bring in the context for the clinician. So the purpose for us is not to determine the diagnosis or predict, but actually to facilitate information for a clinician so that then they can bring in their context towards diagnostic approach.
Great, thank you. And there's another question from Michael, a question with respect to whether there's a preprint. I think there's already a paper, right? It's a Nature paper, is that correct?
Yes, it has been published in Translation Psychiatry. Yes, sorry, I should have mentioned that. The QR code, yeah.
Yeah, great.